Good morning class. Today we will start the chapter Genomics, Proteomics and Bioinformatics. Now can you tell me what do you mean by genomics, proteomics and bioinformatics? Genomics. What is a genome? Genome means the total DNA complement of a cell. So genome means the total DNA which we study in a cell, and studying that genome is genomics. What is proteomics? Study of protein in a cell is proteomics. And what is bioinformatics? Bioinformatics is when we use computers, information system for the storage, retrieval, distribution, sharing of information which we generate in biology. So this chapter talks about finding out the information and then storing the information for sharing with other researchers, other collaborators. So term genomics, it was coined by Thomas Roeder in 1986. Genomics is the scientific discipline of mapping, sequencing and analyzing genomes. Now what is the difference between sequencing, analyzing, mapping? Any difference? Sequencing. What do you mean by sequencing? Sequencing means, as you know, a particular chromosome is a stretch of DNA, or DNA consists of chromosomal stretch. So when we see that stretch, it is composed of nucleotides. In case of DNA, A, T, G, C, 4 nucleotides consist, or 4 nucleotides join together to form a chain which is DNA. DNA then condenses to form chromosome. So when we try to find out with the help of experiments, when we try to find out the sequence, that is ATGC sequence, in what sequence does the nucleotide combine? All the nucleotides, in which sequence do they combine to form the DNA? That is known as sequencing. One of the methods of sequencing DNA is Sanger's dideoxy method, and second is Maxim-Gilbert. But in your class, we'll be studying about Sanger's dideoxy. Now what is mapping? Mapping means finding out the positions or locations of different genes which are present in the chromosome. So mapping is finding out the position, like you create geographical maps. Similarly, we create maps for the DNA, for the chromosome. So mapping is finding out the location of different genes in a chromosome, and sequencing is finding out the sequence of nucleotides present in the genes or the chromosome. And then analyzing. Analyzing means the genome sequencing and mapping. Once it is done, now we need to find out what it codes for. What is its function? What is its role in the particular organism from which it has been isolated? H. Winkler in 1920 coined the term genome. Genome is a complete set of chromosomal and extrachromosomal genes of an organism, a cell, an organelle or a virus. What is bioinformatics? Bioinformatics is the management and analysis of biological information which is stored in the databases. Human Genome Project. In the United States, Human Genome Project officially started on 1st October 1990. It was a 15-year program. This program aimed to sequence the complete genome of the human being. Now why do we need to sequence the genome? Why do we need to undertake such a tedious task? Let the things be as it is. We have been surviving since time immemorial and we can continue surviving. What is the need to sequence the genome? Genome sequencing helps to find out the genes as well as the sequence present in the genes. And then this helps to find out what mutations occur in case of different kinds of diseases, different kinds of malfunctioning of the body, the reason behind the malfunctioning, the reason behind the diseases, reason behind the genetic diseases, reason behind inheritance, inheritance patterns, DNA fingerprinting. All this can be done only after we can find out the sequence of the genome. So, genome sequencing project had multiple aims. Now, as we discuss, what were the goals of Human Genome Project? To map and sequence the complete set of human chromosomes as well as those of several model organisms. To sequence an estimated 3 billion base pairs of the human genome. To make a series of maps of each human chromosome at increasingly finer resolutions so that we know all the integrities of inheritance. J. Craig Venter, a researcher at the National Institutes of Health, devised a new way to find the genes. And what was the new way? He found out about expressed sequence tags. What are expressed sequence tags? As the name suggests, these are the sequences present in the genome which are expressed. Now these expressed sequences, portions of these expressed sequences are taken as tags. They are used as tags to locate other genes. Isolation of messenger RNA molecules. The first step in creation of express sequence tags is isolation of messenger RNA molecules. The second step is copying these RNA molecules into DNA. Now, you must have heard about central dogma in molecular biology. What is the central dogma of molecular biology? The central dogma says DNA forms RNA, RNA forms protein. It cannot go in the reverse direction. But these days, a twist in the central dogma, a modification, an addition has been discovered which says RNA can be reverse transcribed to form DNA. Since formation of RNA from DNA is known as transcription, so the reverse formation of DNA from RNA is known as reverse transcription. So, copying these RNA molecules into DNA, now the DNA which is formed from RNA is known as cDNA. Sequencing a part of these DNA molecules to create express sequence tags or ESTs. In short, express sequence tags are known as ESTs. So the cDNA which is created, since it is formed out of the mRNA, mRNA is a product of expression of DNA, that is why we name it as express sequence tags. What are the applications of ESTs? They are used as handles to isolate the entire gene. Genes which are present in different colonies of bacteria. When we create libraries, genomic libraries and cDNA libraries, in the libraries we need certain probes, we need certain handles to find out the gene of interest. So ESTs help us do it. ESTs help us find out the desired DNA, where it is present, in which colony it is present. It represents the active portion of the genome which was producing messenger RNA for protein synthesis, and hence we name as express sequence, since it was the active portion and it was expressing itself to produce messenger RNA. Then the EST approach has generated enormous sized databases of nucleotide sequences. Creating ESTs, finding out ESTs, it has created enormous sized databases. That means every day we are discovering n number of ESTs, and these ESTs, the information related to ESTs, it has been put on different databases so that it can be accessed by any researcher, anybody around the world. This approach has facilitated construction of preliminary transcript map of human genome. So transcription means formation of RNA from DNA. So when we take RNA, convert it into DNA, that is cDNA, and then we use it to create a map, that becomes the transcript map, since we are using the product of transcription. The development of EST technique is considered to have demonstrated the feasibility of high throughput gene discovery, screening of all possible gene candidates from the EST library, as well as provided key impetus for the growth of genomics. Now we come to genome comparison. The 0.2% difference in DNA sequence is enough to make each individual unique. So, it is understood that on an average, 1 in 1,000 nucleotides are different in genomes of two different individuals. Since we have approximately 3 into 10 to the power 9 nucleotide and base pairs in a human genome, so if 1 in 1,000 nucleotides are different, imagine the enormous difference present between two individuals. What are SNPs? SNPs are single nucleotide polymorphism. What do you mean by polymorphism? Morphism means many forms. Poly means many. Morphism means form. Poly means many. So, single nucleotide polymorphism means many forms existing in nucleotides. What do you mean by that? You studied about codons. What are codons? Codons are triplet nucleotide sequence which are coding for a particular amino acid. So, for a particular amino acid, we have multiple codons except few. Among these multiple codons, which codon is present in which individual depends on family inheritance pattern. So, when we see the enormous diversity that is created due to permutation combination of different codons in different individuals, it leads to presence of single nucleotide polymorphism. So, SNPs can occur both in coding and non-coding regions of the genome. SNPs are DNA sequence variations which occur when a single base, that is A, C, G or T, is altered, so that different individuals may have different bases at these positions. It is believed that SNPs occur at 1.6 million to 3.2 million sites in the human genome. SNPs may affect gene function depending upon exact base change and where it occurs. The variations between individuals, that is particularly in the non-coding parts of the genome, are exploited in DNA fingerprinting, which is used in forensic sciences. What is DNA fingerprinting? In case of DNA fingerprinting, we use restriction enzymes to cleave or cut the DNA from different individuals. Suppose I have a crime scene. At the crime scene, I find a DNA sample. Now, I doubt this particular crime must have been committed by any one of the two suspects. How do I find out which one is the criminal out of the two suspects? So we go for DNA fingerprinting. This is the use of DNA fingerprinting in forensic sciences. Now, a single base difference in the ApoE gene is associated with Alzheimer's disease. Until and unless we study single nucleotide polymorphism, we will not be able to find out which particular mutation, which particular variation is responsible for a disease. Here we give an example of ApoE gene, which is associated with Alzheimer's disease. So it is possible to know only after we sequence the genome. A simple deletion within the chemokine receptor gene, that is CCR5, leads to resistance to HIV, that is human immunodeficiency virus infections, and the development of AIDS, that is acquired immunodeficiency syndrome. Again, this is possible only after we sequence the genome. In SNP analysis, it is important. SNP analysis is mainly important for diagnostics purpose. What do you mean by diagnostics? Diagnosing a disease. So physicians can use SNP map to determine how the patients are likely to respond to a particular drug. So it has a dual purpose here. It can be used for diagnosis of a disease. Also, it can be used to find out the diseased person, that is the patient, which particular medicine is the patient going to respond to. Since the reasons behind particular disease will be known by sequencing. So when we know the reason behind the disease, it would be very easy to cure or treat that disease. SNP analysis can be used in population genetics, as some SNPs vary in different frequencies between populations. So we can study different populations, the similarity, dissimilarity between populations. Comparative genomics. Now when we study genomics, we can compare different organisms. We can compare different complement of DNA. We can compare different RNA present at different developmental stages, in different cells of the same body, in different organs of the same body. So comparative genomics, it is estimated that the difference between human and chimpanzee genome is only 1 to 3%, while human and mouse share about 97.5% of their working DNA. These similarities suggest that none of these genomes has changed much since we shared a common ancestor 100 million years ago. Looking at the morphology and anatomy does not give us such in-depth analysis of similarity of different species, different organisms existing at different hierarchy in evolution. Only genomics, genome analysis, comparing different genomes, it can help us find out the similarity, dissimilarity. It can also help us to find out the evolution, the phylogenetic relationships. Now we come to microarray technology. Comparative genomics uses a technology known as microarray technology. What is a microarray technology? Micro, as the name suggests, small. Array, a sequence, an organized sequence of something. So what is organized sequence here? In case of microarray technology, in case of biotechnology, we use genetic material to be on the microarray slides. The genetic material might be DNA or RNA. So DNA or RNA can be put on the slides. It can be etched on the slides, and these slides, which are made up of either glass or silicon, they can be used for comparing genomes, comparing DNA, comparing RNA of different organisms, or maybe of the same organism but of different organs, different cells, comparing at different developmental stages, childhood, adolescent, old age, etc. This technology promises to monitor the whole genome on a single chip so that researchers can have a better picture of interactions among thousands of genes simultaneously. The biggest benefit of microarray technology is we can study multiple genomes, multiple genes at the same time on a single chip. So making our experiment a bit less tedious. Studying each and every individual gene separately in separate experiments lead to wastage of time. A lot of time is required, a lot of money goes into it. So microarray helps us to cut down on time and money. Microarrays consist of large number of DNA molecules spotted in a systematic order on a solid substrate, usually a slide. The base pairing or hybridization is the underlying principle of DNA microarray. Microarrays exploit preferential binding of complementary single-stranded nucleic acid. Now what do we mean by hybridization? When two DNA fragments or one DNA, one RNA fragment, they are single-stranded and they're made to find out the complementary part and bind to it, forming hydrogen bonds. That is, if on a microarray chip I have spotted single-stranded DNA of human, and then I use single-stranded DNA from another human to find out which genes are similar, which genes are different. Then the similar ones having complementary sequences, they're going to find out each other and bind to each other, forming hydrogen bonds. This is hybridization. So hybridization is the underlying principle of DNA microarrays. The type of molecule placed on the array units varies according to circumstances. The most commonly used molecule is cDNA, that is complementary DNA. As we just discussed, complementary DNA is a DNA which is formed by reverse transcription of RNA, or complementary DNA which is derived from messenger RNA. Since cDNA are derived from direct messenger RNA, each cDNA represents an expressed gene. In order to detect cDNA bound to the microarray, they must be labeled with a reporter molecule that identifies their presence. What are the reporter molecules that we can use? It can be fluorescence, it can be radioactivity. We can use, preferably we use fluorescence in case of microarrays, like green color fluorescence, red color fluorescence. These fluorescent molecules can be combined with the DNA fragment, so that when the DNA fragment goes and binds to its complementary sequence, we can locate it by seeing the fluorescence. Now we come to another technique, which is fluorescence in situ hybridization. As the name tells you, here also we use fluorescence. Again, here also we use the principle of hybridization. It is possible to introduce colors into DNA by a technique called Nick translation, developed in 1977 by Whitby and Paulberg. Now what is Nick translation? Nick translation is when we take the original DNA sequence, we nick it. Nick means put cuts, cleave it at some points, so that the new DNA that is formed by copying the template incorporates the newly provided nucleotides, and the newly provided nucleotides are the labeled fluorescent nucleotides. So the newly formed DNA fragment has label. The enzymes DNA polymerase 1 makes DNA and DNase 1 cuts DNA are combined in a buffered reaction with DNTPs, that is deoxynucleotides, including DUTP labeled with a red or green fluorescence. The DNA polymerase 1 adds fluorescence labeled nucleotide residues to the 3' hydroxyl terminus, that is the result of nicks created by DNA1 in the DNA. The DNA fragment size with fluorescent probe after nick translation depends upon the amount of enzyme and the incubation time of the reaction. The size can range from 300 to 3000 base pairs. The more time we give for incubation, the longer is the fluorescence DNA formed. Now this technique, it is used for diagnosis and monitoring of treatment of chronic myelogenous leukemia. In short, we call it as CML. Karyotype analysis of the lymphocytes made from blood samples of CML patients show 922 translocation in the chromosome, that is Philadelphia chromosome. What is Philadelphia chromosome? You must have read that in case of meiosis, the homologous chromosomes, they pair up and they exchange segments. That is simple crossing over, and that creates variation, but that is normal. When the crossing over or the exchange of fragments takes place between non-homologous chromosomes, here in this example it is taking place between 9 and 22. So it becomes a reason for a disease. So Philadelphia chromosome leads to chronic myelogenous leukemia. By counting the number of such cells, it is possible to find out severity of the disease. The more the number of cells having Philadelphia chromosome, the more the severe is the disease. From the DNA library, it clones carrying the particular genes involved in CML are picked up. Using nick translation, it is now possible to fluorescently label chromosome 9 region with red color, chromosome 22 region with green color, and prepare the probe. Now, reciprocal translocation between chromosome 9 and 22 forms an extra long chromosome 9, that is DER9, and Philadelphia chromosome, that is PH1, containing the fused ABL BCR gene. When CML lymphocytes smear cells are hybridized with the two probes, that is the 9th one having red color and 22nd one having green color in situ, and when observed under fluorescent microscope, the cells which are affected appear yellow. That is because of mixing of green and red color in fluorescence produces yellow color. The unaffected cells, they appear separate as red and green. Fluorescence in situ hybridization, in short, we call it as FISH. It allows knowing the status in the interphase, unlike in karyotyping, where we need a metaphase chromosomes. It is possible to monitor the effect of chemotherapy and the drugs by taking out samples and counting number of cells appearing yellow. So students, for the class, we stop here today. The rest of the chapter we'll discuss tomorrow. Thank you.